Right, so good afternoon, everybody. I'm Simon Kerr, CEO of Cypralis. I'd like to thank the organisers for the opportunity to present to this small but august group this afternoon. In the next few minutes, I'm going to present the Cypralis strategy and our capabilities regarding drug discovery and development of novel cyclophilin inhibitors for the treatment of acute and chronic degenerative diseases. So, Cypralis is a Cambridge, UK-based biopharma company focused on discovering and developing novel cyclophilin inhibitors as first-in-class disease-modifying treatments for degenerative diseases. We spun out from a UK-based CRO, Celsia, back in 2013, with a mature cyclophilin inhibitor discovery platform already in place, and we've raised about £4 million to date. We retain a close link to Celsia, which allows us to operate with a lean, cost-effective outsourcing model. And we have exclusive access to the world-leading cyclophilin chemistry capabilities that Celsia built up prior to the formation of Cypralis. We're looking to raise approximately £8 million during 2018 to build a pipeline of preclinical candidates that inhibit cyclophilins to treat chronic diseases in three areas: fibrosis, neurodegeneration, and a novel approach to T-cell exhaustion, which will be placed into asset-focused IP companies that can be exited on a one-by-one basis. So to introduce cyclophilins as drug targets, these are enzymes with well-characterized active sites, and they're assayable using binding or functional methods. There are several subtypes that have different functions, and the general interest in cyclophilins as a class of inhibitors targets has been growing for several years. However, the existing inhibitors, of which cyclosporin A is the best known and most widely used, are non-selective and cause immunosuppression via their interaction with calcineurin. They also have poor drug properties. So there's a clear need for a new generation of inhibitors that are not only non-immunosuppressive, but that also offer subtype selectivity, have good drug-like properties, and can be formulated via different modalities, including IV, oral, and topical. Cyclophilins have been well validated in several diseases by knockout, RNA silencing, and from clinical data obtained with cyclosporin and the second generation inhibitors, including alosporavir and SCY-635. So the inhibition of cyclophilin has been implicated in several major therapeutic areas. We're focusing on three subtypes: cyclophilin A, which is a secreted cytokine; cyclophilin B, which sits on the endoplasmic reticulum; and cyclophilin D, which is resident on the inner mitochondrial membrane and is a well-validated component of the mitochondrial permeability transition pore, or MPTP. There's a significant body of evidence that's been generated over the last few years that supports a role for the opening of the MPTP as a gating event for cell death following oxidative or other stress insults to a cell. And this event has been shown to be common to several disparate conditions, including ischemia, reperfusion injury, certain muscular dystrophies, tissue injury resulting from excessive inflammation, and neuronal cell death. And in turn, cyclophilin D blockade has been shown by several groups, including our collaborators at the University of Liverpool, to be effective in protecting animal models in several diseases, including Parkinson's and ALS. So cyclophilin inhibition opens up a novel approach for several degenerative diseases, including the areas that are shown on this slide. So in terms of strategy and our platform capability, first of all, we have a powerful discovery platform with access to around 30 biochemical and functional assays covering the major cyclophilin subtypes, as well as other isomerases, including FK binding proteins and parvalins. We own over 1,800 novel designed cyclophilin inhibitors and data on them that span three major chemical families and classes, including cyclosporin analogues, peptides, and then an entirely synthetic class of new cyclophilin inhibitors, which we've designated as the Cypralides. We believe this represents the largest and most structurally diverse library of cyclophilin inhibitors that exist, and what particularly discriminates and distinguishes our library from other approaches is the presence of compounds which for the first time provide subtype selectivity to cyclophilins A, B, and D, and that provides the basis for several different TPPs. It's a highly drug-like collection. It includes compounds with excellent PK properties, and we have the potential for delivery by oral, IV, and topical routes. We also have the first cyclophilin inhibitors that are brain penetrant, and that offers a potential breakthrough in neurodegenerative diseases. So the role of cyclophilins in various pathologies opens up a broad set of diseases that we could potentially approach, ranging from acute to chronic and from peripheral tissue to the CNS. As a small company, it's attractive to us to pursue indications ourselves that require short duration of treatment as a pathway to an early clinical proof of concept. And what we're doing in our most advanced program is in acute kidney injury, where we're taking that forward through a Chinese partnership, and we'll be filing for an IND at the end of, our partner will be filing for an IND towards the end of this year before taking that program into clinical development. However, the main thrust of our Series A plan is to generate clinical candidates for chronic diseases, including fibrosis, neurodegeneration, and IO. And these are all areas in which the industry is clearly seeking novel disease-modifying candidates. We can see that companies are prepared to actually enter into deals with very substantial upfronts and even larger milestones if there's the basis for modifying the outcome of a previously untreated condition. Thinking there of the kind of deals that Nimbus have done with Gilead in phase 1 in fibrosis. Takeda have just transacted with Denali at a preclinical stage to get their antibodies into the brain. So we think the value proposition for a disease-modifying approach in these indications is very clear. So our most advanced compound is being taken forward in acute kidney injury, where cyclophilin D knockout and or inhibition has been shown to be highly protective in animal models of AKI and also of acute pancreatitis. We've replicated this work using our own compounds in vivo and with profound protection of tissue injury driven by various toxic insults, including ischemic injury, bile salt, and endotoxins. We've signed a deal with Farsight Shanghai, which is currently taking CC4066, our most advanced compound, into and through IND-enabling studies. And as I said, that should be filed with the Chinese FDA before the end of 2018. It will then go into clinical development in China, but we retain the rights for development of this compound in the West, and we get access to all the data that's actually generated through our Chinese collaborator. Hospital-acquired AKI is a really big problem, and it actually affects 3 million cases in the Europe, US, and Japanese markets. And I think when you look at the mortality, it's really actually very shocking that it represents for nearly 25% of hospital-acquired AKI. The next program is fibrosis, and there's good clinical evidence here that cyclophilin inhibition can result in the protection of the liver from fibrosis subsequent to hepatitis C, and can also improve established fibrosis in the lung. We've found that collagen production by TGF-beta stimulated hepatic stellate cells can be potentially inhibited by our cyclophilin inhibitors, and that could in turn provide a treatment for ongoing fibrosis. We're currently screening our library for compounds with dual activity, an anti-collagen and a cell protection phenotype, which if successful will take into various models of fibrosis. We then have the basis for executing a lead optimization program with the objective of selecting a clinical candidate for liver fibrosis subsequent to NASH, which is clearly a major area of interest for the pharma industry. At the same time, there are many papers which point to the role of cyclophilin D knockout as being protective in several models of neurodegeneration, including the mutant alpha-synuclein model of Parkinson's, the ALS SOD1 model, and the APP Alzheimer's mouse models. We've been working until recently with Alzheimer's Drug Discovery Foundation in New York, who provided us a grant award to develop cyclophilin D inhibitors for neurodegenerative diseases. And we're very excited to have actually generated the first cyclophilin inhibitors that cross the blood-brain barrier and achieve very high brain penetration following oral dosing in 2 species. We're currently running an in vivo neurodegeneration model in Israel that will report out in the next couple of months. And we then plan to run a lead optimization program as part of our Series A fundraise to generate a clinical candidate, as well as at the same time generating data from human iPS cells and building new collaborations with leading academic groups. At the point of clinical candidate, we'd then be looking to establish a partnership with a large pharma or biotech company for onward development. The third optimization program we are looking to run will be in immuno-oncology. Cypralis owns compounds that have shown the restoration of immune responses in a virology indication and have reactivated T cells consistent with the reversal of a T cell exhaustion phenotype. This is a very interesting profile, we believe, and we're evaluating several compounds from our library in models of T cell exhaustion and adaptive immunity. If we replicate our previous data, this will provide the basis for a lead-op program to generate a clinical candidate, and the plan would then be to collaborate with leaders in the IO field with the goal of augmenting checkpoint inhibitors by the addition of a new immune-boosting mechanism. Conscious of time, and in terms of the leadership team, it's very small, but we're very highly experienced, and with a range of capabilities that have been built from our prior careers in big pharma and biotech. I've worked in the industry in commercial leadership roles for around 30 years now. Our chairman has been involved in the cyclophilin field going all the way back to Sandoz, Novartis. And our CSO, Mike Peel, who's in the room, has similar experience from Glaxo and his previous company, Synexis. We have a network of excellent consultants and scientific advisors who bring therapeutic area expertise, and our plan is to continue to build out our network of external collaborators and advisors whilst retaining a core leadership team. I'd also quickly like to mention our existing partnerships with Celsia, from which we span out, Gilead Sciences, Cedars-Sinai UCLA, the University of Liverpool, the University of Cambridge, and also to note that we've received, and are very proud to have received 3 awards actually from Innovate UK under the Biomedical Catalyst scheme. We also, and somewhat remarkably for a company of our size, have a very well-established set of collaborations in China. And we're working with Farsight Shanghai, who I've mentioned previously, Fudan University, Shanghai Jiaotong University, and the West China Hospital, which actually opens up access to some excellent science and some very substantial grant and industry funding as a very interesting route for us. So to summarize, Cypralis has a powerful, and we think it is an unrivaled cyclophilin inhibitor platform with the potential for generating first-in-class drugs to treat several acute and chronic degenerative diseases. Our Series A business plan is to generate clinical candidates in 3 programs, each in areas of high interest for major pharma companies. We're seeking to close a Series A round of 8 million sterling by mid-year, and we're currently seeking term sheets from potential lead investors. We have an electronic data room accessible under a CDA, and I'd be delighted to talk to people afterwards about follow-up, etc. So I stand between you and lunch, and on that basis, I'll just say thank you and take any quick questions.